If you're doing a chemical or mineral test, you can wait. But if you're testing living things, you want to treat them like living things, right? You don't catch a fish and throw it in the back of your truck for a week and then weigh it, right? And then certainly not eat it. So we say for our test, go get field moist fresh soil from the rhizosphere, test it right away. Don't let it dry out. Don't abuse it, right? Don't mix it up.
Soil testing doesn't have to be overwhelming or daunting. It can actually be simple, inspiring, and bring curiosity and wonder into your farming and gardening practices. Laura Decker from Prolific Earth Sciences is going to give us a rundown of different soil testing that's been around for conventional or chemical farming, and also what's being used more in the regenerative, organic, and biodynamic spaces, and how we can learn from them and what we learn from them, and maybe what soil testing is best for you and what you want to do. I'm excited to bring this conversation with you. Prolific Earth Sciences is also the producer of the MicroBIOMETER, which is a soil test that tests the fungal to bacterial ratio. So you'll get to learn more about that as well and how it works. Enjoy the conversation with Laura, and you make yourself an amazing day. Thanks for joining us for the Soil Summit 2024. Biodiversity, cultivating life. It's awesome to be in the room with you again, Laura, to talk about soil testing and all things soil testing. And you're probably one of the best people to talk about the different types of soil testing with, for lots of different reasons. Your mom was a researcher, and you have that kind of curiosity in your genes, and you actually have the microbiometer, and you have a depth and breadth about soil testing that I just thought would be really valuable, or we just thought would be really valuable for people to learn, that there's not just one way to test your soil, and there's lots of different ways to test your soil, and there's lots of different reasons you might want to consider different soil tests. So thanks for joining us. And
Yeah. Well, thank you. Thank you. Thank you. I'll let you know that I know a lot, but not everything. So it'd be nice to hear feedback from all the people who do soil testing. And we'll sort of talk about soil testing in general. You can jump in. But what I will say to all the growers out there is you need a lot of information to make decisions. So there's not really, as much as I would love to have the microbiometer be the only thing you ever need, that is not true. You will need lots of different measurements. The good news is a lot of them are very cheap, very accessible, or you can just sort of do them yourself. So it's not particularly difficult. Don't feel like if you're getting into soil testing, you need to spend, $300 on a lab test. You can if you want, but you don't have to.
That's encouraging, because sometimes the barrier of entry
Yes.
feels very, very big, and it doesn't have to be. So I like that you said that.
Yeah, and it's better for people to just sort of start at the basics, and then as they get to sort of digest more information and be able to act on it, then they can add more metrics. But don't start getting 10,000 data points if you don't really know what to do with them, because data is only as good as it informs your decisions. So think about, what decisions do I have to make, and what would help me make them?
Yeah, and I'm going to echo that again, because what you said is so powerful, was do what you can take action on.
Yeah.
Right? Do what you can take action on. And unless you're a curious soul with a disposable income that just likes to gather data, because that might be your form of action, is gathering information. So also just being really self-aware of where you're at on your journey is important.
Yeah, good. Well, you want me to share my screen?
Share your screen. Let's
Great.
chat about soil testing.
Good. So this is the basics of soil testing. There might be people out there who know more than me. But hopefully this will be a good level set discussion. So the first thing we're going to do is talk just sort of in general a little bit about regenerative farming. So I'm sure we're sort of all on the same team, but over the past decades, farming has relied on both chemical input and chemical testing, and what I like to call chemical farming, right? The focus on chemical inputs has damaged microbial communities and damaged the relationship between plants and microbes. So in current conventional farming, you put down seeds and you throw down chemical fertilizers that the plant uptakes, right? Sort of in a more natural relationship, microbes and plants have a symbiotic relationship. They feed each other, they barter. It's a really cool system. And the microbes make things bioavailable to the plants that the plants can't access without the microbes. So there could be tons and tons of, say, phosphorus in the soil, but without microbes to make it bioavailable, the plants can't get it. So if you are doing chemical farming, you just have to throw more and more in, right, until it's there. The other things is plants are very, very efficient. If they don't have to barter with microbes for nutrients, they won't, which means they won't feed the microbes, which means the microbial community will go down. So one of the results of over-fertilization is a dwindling of the microbes because the plant doesn't need that. That is bad because now you have crops reliant on very expensive, dangerous, often environmentally harmful nutrients, but a lot, and we're learning more and more, a lot of the nutrient density and the micronutrients in plants comes from microbes. So if you don't have that, you don't have those micronutrients, you don't have all the antioxidants, which is why people like organic food better. I know sort of the institutions tell us there's no difference, but anyone who eats an organic diet will tell you it tastes better and I feel better. And it's in part because of that. Also because it's not covered in chemicals that you're ingesting. So when we think about switching from chemical farming, chemical inputs, right, into more biological, we think about testing. Well, what do I need to do to make that transition, right? Again, healthy soil is dependent upon the microbial community for minerals and nutrients, digestion of litter. So as, say, you have a cover crop growing and you have roots underground, right? If you let those die, the litter will decompose, being carbon for the next generation of plants and microbes. And the roots will decompose, both leaving organic matter, but also leaving the little holes where oxygen and water can get in, okay? Which is soil structure. Soil structure is created a lot of different ways, but really good soil structure increases your water holding capacity, increases the aeration of your soil and protects from erosion. And microbes and plants give each other information about the environment, what pests are coming, what's happening with drought, and they adapt and protect themselves. And then, of course, sequestering carbon. So we know that if you leave litter and organic matter into the soil, right, it not only creates a good environment for next year's microbes to grow, but it does sequester carbon. So that's one of the things around the world that particularly smallhold farmers are looking to do is get paid for carbon farming in addition to crops. So a goal, building a balanced microbial community is really hard. And that's why all your users are relying on you for information. The goal is to create optimal conditions for microbes to thrive so that your plants will thrive. The microbial community can do this if it's given the right foods. And feeding microbes enables you to jumpstart the whole system. We call it a virtuous cycle, right? Microbes grow, plants grow. The more plants there are, the more microbes there are. Then that's more soil organic carbon in the soil, which is food for next year. So we think of that as the virtuous cycle and the chemicals as the vicious cycle. More chemicals, fewer microbes. Fewer microbes need more chemicals, right? And on we go. So when you think about, here's my soil, here's my crops, what I'm going to do, right? And we'll get to testing in a minute. You think chemical fertilizers, right? The standard ones. We know that they're expensive. Over time, they deplete the population. But we do know that sometimes with judicial use, they are important, right? Microbial inputs, mycorrhizal fungi, all kinds of inoculants. Sometimes they're helpful, sometimes they're not. Depends on a lot of things. There's some amazing products out there. Anecdotally, from people, we find out that the best products are the ones that feed the existing microbes, not necessarily microbial inputs, although sometimes those are necessary, but it's better to have something that is something that you can sort of prime the microbial community to get going, okay? And research is ongoing. It's unclear if one microbe is the same in all kinds of places. It's just there's so many millions and billions of microbes, it's hard to know. Cover crops, that's kind of a mantra on a standing thing, no bare soil, right? You want roots growing, you want roots in the soil, you want the ground covered for heat, insulation against cold and hot, hold water, keep roots in, keep things growing, keep the microbe community even in the winter a little bit dormant, and biostimulants. Okay, biostimulants, another thing that are getting very big and regenerative that can stimulate both microbes and roots to grow. So now we get to testing. So you sort of know, right? Everyone has a plot of land, right? They know I want it to be more productive. I'm rehabbing it. What do I do? Right? What inoculants, what fertilizers are the right thing for me to do? So the traditional tests out there are NPK, nitrogen, phosphorus, and potassium, right? Those are sort of the big three in agriculture. They really spurned the Green Revolution in the 60s to increase production. It was very effective, but now we're sort of dealing with the unintended consequences of it. Sometimes those are still really important to do. What I will say in particular for potassium is a lot of potassium can be made bioavailable by microbes. So even if you have low potassium, you could have a microbial community that is feeding enough potassium to you. Same with phosphorus and nitrogen. It's a little bit hard to know, right? But if you get a report back, that says you're low on something, don't automatically think you have to add it or think you have to add it in a chemical way, right? You could add some compost, or other inoculants. Another test is pH. pH is testing for soil acidity and basicness. pH testing, there's a billion meters out there. They're all very controversial. And my husband works in horticulture, and he says, you calibrate them and they get all wrong. So pH, while it seems like it should be sort of an easy, quick thing to do, is a little bit more complex. Soil pH is sort of a condition that's hard to remedy, right? You can put down a couple things to make it more basic and acidic, but just be aware that there is some variability in all the tools and all the testing of pH. And Natalie, you might know a little bit more about the complexity of pH testing. Another thing is moisture, and moisture, water infiltration, you can do for free with, a piece of plastic wrap and a little ring. You can go on YouTube. What you want to say is not how moist, well, you can figure out how moist is my soil today, but water holding capacity is the other thing. How quickly does my soil absorb the water? So if I get a big rain, is it banking all that water, or is it staying on the surface and running off and not doing me any good, right? So moisture is both sort of standard moisture of a given thing, but also water holding capacity. Those are a little bit traditional. There's a lot more. Now that you bring some up. In regenerative testing, right, we add more biologics and more soil structure discussion. So microbial biomass, right? Where are my microbial communities going? What's my fungal to bacteria content? People are very interested in fungal content. It grows soil structure much better. And for food that you eat, it does a lot for taste, just like wine is in the terroir. So is other foods. People care a lot about the fungal content. And fungal content is sort of the easiest to destroy with tilling, right? You just smash them all up and they're gone. Microbes are so little, they can survive a lot more. Water holding capacity, we talked about. CEC, which is cation exchange capacity. I don't know a whole lot about it, but I know that a lot of people use it, and it is useful for them. BRICS, which is a test sort of the nutrient density of leaves. And again, that's a meter that people get. It's still early on in the testing, so I can't really speak to how useful it is, but a lot of people do use it. Nutrient density testing is another thing that's getting really big, right? You're saying if I have really healthy soil, really good microbes, really good conditions, my plants will have more antioxidants, right? More micronutrients. And can I prove that out, that sort of my spinach is much hardier than other people's spinach? Sap testing is another thing that people are moving into, and that is taking the sap from the leaves and testing it for all kinds of nutrients. And that really gets to the fact that even if my soil on paper doesn't look good, but my plants are doing terrific and they are, thriving, you maybe don't have to do anything about that. So Natalie, do you want to throw in some more examples of
Yeah.
testing and how people use them?
Yeah, there's the pedometer test. I think that's what it's called. It's the testing the like basically how the compaction of your soil, how
Oh.
deep, like how soft or hard your soil is. And that's a really interesting test. And then there's also the testing the name just escapes me. The the electromagnetic energy in your soil. So
Mm-hmm.
that there's some really cool tests. You can actually just if you dig a hole and you have a plant with roots on it and you're putting the plant into that hole, you might notice the plant's roots are attracted to the edges of the
Oh,
hole that you just dug, and that is the energy, that's electromagnetic energy that's in your soil. So there's actual instruments that'll test that too, and you want soil that is healthy and has energy moving in it.
yeah. Good. And then there's lots of tests of just feeling the soil, smelling it, right?
Mhm.
Looking at the like you have a lot of clumpy soil, which is a sign of soil structure and little homes that microbes make. Um, so a lot of it is looking at it. And then there's also, it's not testing, but there's a lot of information now about what kind of soil you have, right? Where am I geographically? Is my soil heavy in clay, sand, very loamy? And that stuff is now becoming more readily available to people for free to sort of say, you know, I'm growing outside. I'm not going to, have containers full of soil that I sort of make or compost, or maybe you will, but you have to know that you're in a place and you want to use the real soil and make it as healthy as you can.
Mhm.
So it's good to get some data about really what's it like to begin with. Um, particularly with people with a lot of clay. I'm in the Pacific Northwest. We have a lot of organic matter in our soil, but in places we have a lot of clay, and that creates, some difficulty
Mhm.
at times.
Yeah, right. And then there's the fun test of wearing underwear and seeing how long they take to decompose, right? Or the tea bag that you were mentioning.
Yeah, and that is sort of a test of how how quickly your soil is churning, right? The cycle. Like, if you had very dead soil and you put underwear in it. For those of you who don't know, you take usually in two different places, a pair of sort of briefs, bury them, dig them up after a period of time. How I guess it depends on how long.
Yeah, I mean, you can do it weeks, you can do months, and
Yeah.
it
And
depends how active your soil is, right?
yeah.
Because you can if your soil is super active, you can actually see breakdown starting to happen quite quickly. But if I think I wish I looked it up before we hopped on this
Yeah.
call.
And they're fun
Like
online. I will tell you, make sure you get cotton underwear. You don't want to
that's
bury
right.
the polyester in the ground. That's a different test. But then you can see like, oh, this soil that is healthier and has microbes, they're eating stuff, churning it over. The microbes are reproducing very quickly, and consuming everything. And that would be a good test of when I put down a cover crop, when are all those roots, right, turned into stable organic matter? How long does that take? Or is the soil sort of dead? And if,
Mm.
like, for example, if I put down a bunch of compost and cover soil over dead soil and don't do anything else, will it even work, or will it not sort of be enough to do that? A tea bag is another one. They decompose a little faster, and I know for school kids they can, do them in pots too inside, to see where they go. But I'm sure school kids think underwear is much funnier, so
Yeah.
you might
Yeah.
want to do that. And another thing people do is they measure the difference between the temperature in the air, right? Particularly if you're in somewhere hot, and the temperature just under the, if you have some cover or some mushed down crops in your garden. If it's hot, you want to say, okay, I have a big differential between the air temperature and my soil temperature, and that's good, right? Because things aren't going to dry out as fast and go from there. So those are sort of some general testing you could do. Most of these things you can get tests for yourself. I know Luster Leaf is a company that makes sort of at-home tests. Again, they won't be quite as accurate as a lab test, but they're also not, hundreds of dollars. We have a test, of course, MicroBIOMETER has a test for soil microbes and fungal to bacteria that's done on field moist, fresh soil, right? To get a picture of what are my living and dormant microbes now. But there's also, I'm going to go on, in terms of microbial testing, there's different ones. So our test is a test that's designed to essentially mimic microscopy. So if you have a microscope, if you're in the food soil web, great, it's terrific. If that's not in your skill set, our test sort of mimics that to get a sense of how populous is your microbial community. The other big test for soil microbes is the PLFA soil test. That's a lab-based test. You send the soil in, okay, and they explode the microbes, and look at what proteins and chemical compounds are in there, okay. It takes dry soil and then re-wets it, okay, and extracts things from it. It has weaknesses, but it's a useful test. The weakness is you send it away for a while, it's $60. Another very popular test is the Solvita respiration CO2 burst test. What these all measure, and you can do them yourself, they have kits, is it measures the like you breathing, right? The release of carbon from the microbes. So it's a measure of microbial activity, right? Again, it takes soil, it dries it, reconstitutes it, and then measures what activity the microbes are doing. In our experience and sort of in our logic, taking living things and drying them out and reconstituting them to start reproducing again isn't always the same population, right? Or we don't quite know if it's the same population. We know enough about microbes to know that they are symbiotic. So microbe A might come first, and then it'll make a compound that microbe B needs. So microbe B will start reproducing. Microbe B will make a compound that microbe C needs, right? So it goes on through. These microbes only make a couple things because they don't have a lot of DNA. So they need each other to trade back and forth, right? As humans, we can make all kinds of things in our bodies, cholesterol, who knows what, I don't even know. But microbes are really dependent upon each other. So you can't really have just one microbe in there, right? You need a lot of them. So when you kill them all off and then you bring back the population, which ones are you bringing back, the early ones or the later ones? But respiration is very widely used. It's very easy to get to do. And then we'll do that. And then genomic testing. So DNA sequencing is pretty cool. They sequenced all of us, so now they've moved on to microbes. There's a couple companies doing it. BioMakers is one of the ones that are early on in not only mapping microbes, but in sort of getting that information in the field. It is very expensive, $300 to $400. And it's early in the process. So what they do is they'll take the microbes and they'll sort of put them in what their algorithms assumes to be functional groups, right? This one nitrogen fixes. This one, does potassium. This one, makes iron bioavailable. All these little groups. So when you get that test back, they'll say things like, you have a lot of nitrogen fixing microbes, but not a lot of what we think are potassium fixing microbes, right? I don't quite know what people do with that information because I don't know how you make a particular kind of microbe be more prevalent in your community. Um, but it's certainly helping on the way to get a more complete picture of what's out there. We'll say that there's millions and millions of different kinds of microbes. Microbes vary tremendously genetically between each other. Um, a microbe can have 20 to 30% genetic difference between two things. And we don't really have any genetic difference that big between any other living creature. So they're quite different, and they mutate a lot. Um, so, it's an ongoing thing.
In fact,
But
mutate fast. Like for us, for humans to experience a change genetically, typically that can take a really, really long time for that adaptation. But with microbes, they're like this, right? They're faster than that. You can't even see it with the naked eye how quickly they can, well, I guess.
yeah, I mean, they reproduce in many, many, many times an hour, right? So it goes very, very quickly, and they can
And
die.
right. And not only are they reproducing, though, they're adapting,
Yeah.
which is mind blowing.
Yeah, I mean, they're amazing creatures. They're much better than we are. So that's one of those things. I don't think it's now big farms are doing it to sort of get a sense for it. In general, with a microbiometer, because we don't give that kind of information, we don't know how we would even get it. The thought is you want a large and diverse microbial community, right? If you have a large community, it will naturally diversify, right? Just like anything in the universe. So the larger it is and the more you support it with conditions and not doing anything basically to destroy it, right? If you go into a forest that no one does something to, it's pretty awesome. So that's where we are in sort of testing soil microbes. And then of course I had to give a plug for our little microbiometer, which is, here's our little kit. And it enables you to test it. It's pretty cheap. The tests run between $13 and $7 so that you can really get a sense of what's going on with your microbial community, and then add that information in to all the other data that you have. So right now, unfortunately, I think a lot of growers are in sort of Excel land, right? There are lots of apps coming out that are really good in terms of managing farms and gardens and homesteads. But some of them are for really big companies, and some of them are a little too limited, and some cost money. So I think in the next 2 or 3 years, it'll really be exciting because there'll be much better tools for people to have an app that has weather and geographical data, mapping data, and you can input your things. I will say, why don't I stop sharing this? One of the things about testing is a lot of the companies that do the testing also make money on inputs and amendments and advising. So, and often they have product lines that could be very good or could not be very good. So, you have to, as all things, make sure when you're getting advice from somebody, do they have another motive, right? Is the answer always use my product or something? And it's more nuanced than that.
Yeah. And just can you give us an idea of how people are using the microbiometer, because it's the fungal to bacterial ratio, right, that we're watching with a microbiometer, and so it's not necessarily a one and done test, and that's sometimes people think, oh, test my soil, and okay, so even if they're doing NPK, they just are like, okay, well, I'm going to do more K or less P or whatever it is.
Yeah.
Then it's one and done, and it's like it's changed, and it's like, what? And so soil is something that's really interesting because it is always changing. And I think that's one of the parts of the microbiometer that I really like, is that it wakes people up to that. It wakes people up to being a part of the soil rather than separate from the soil. So you're actually getting to use it and notice how, like, if I'm gonna let you talk to this, Laura. I'm gonna ask you a question. I'm kind of stealing your show here, but
oh, please.
One of the things I like about it is that you can test your soil at a base point and maybe at the beginning of the season, and then you do a cover, like an early cover crop, and then you can test it after that and see what the impact was or wasn't. and you can do mulching on a different area, and you can do microbes, or maybe you have like an input that you're wanting to, a biological input that you're wanting to test, and so you can actually see the impact of the fungal to bacterial ratio and all these different practices, and you can monitor it, like not monitor it, but you can how do you say, you can witness it, you can see it, you can experience it, rather than guessing, right? Because some people are really good at smelling their soil, like you said, pick it up, smell it, notice what
People eat it, you know.
eat it, taste it, like
Yeah.
and some people are really in touch with that, and other people, it's really, it's foreign to them, and they want to know where to start. And so that's what I like about the microbiometer, is it's like, it is a good place to start. And wouldn't it be interesting to taste your super like acidic soil, or
Yeah.
low.
Yeah. What I will say about the microbiometer is the same thing. It's not a number that you want to get, like, I want to be 400. You want to understand the cycles of your microbes in your soil, right? They have a natural pattern, winter, spring. We're talking about a natural process. So it's not, even NPK is not a one and done, right? You should be testing it throughout the year to see if it does changes. pH also, right? Pine needles fall down, it could change the pH of your soil. So especially if you have tools that are low cost and you can do yourself, you can do different testing, because a lot of the testing also, let's be honest, has natural variability. We think as regular people that a test is always 100% accurate. Well, there's lots of different factors in a test. So the more data points you have, the more, maybe if you do say pH, you tested a bunch, maybe there's a couple of strange outlying numbers. If you have enough data points, you can say those were sort of one-offs. They don't really represent the pH of my soil. Same with microbes, right? You want to test in winter, the number will be actually lower, and in spring higher, right? So you want to say, here's my yearly curve. As I do these better practices, right? My curve was like this. Now my curve is a little higher everywhere, right? But it's not always higher all the time, because they have a life cycle, right? Just like people. Populations change and vary. So you do want to have a lot of data points. And the other thing we talk a ton about is changes over time and space, right? You have a field on a given day, you can test 2 points. They'll be different, right? And then if you test this point today and in a month, it'll be different. What you don't want to do is compare 2 different points over 2 different time periods, right? Because you don't know if you're getting time or space variability. So one of the things we encourage people to do is pick a couple spots that you do your testing at all the time, so that you're comparing the same spot over time, or you're comparing at the same time different spots to see what's happening.
That's really good advice. And that is translatable to any soil test, right?
And that is, yes, absolutely. pH, right, will have a natural variability as well. So you don't want to, you could run around and stick it in the soil a billion times and get different numbers. It doesn't mean they're wrong, right? It means they're slightly different.
Yeah, it's curious. And I guess it's because sometimes what people do, they'll get 3 soil samples from different parts of their field, right? And then they'll test their soil. So is that something that someone would do with a microbiometer or not?
So traditional agronomists do composite testing. Okay. So they'll take cores from different places, mash it all together and test it. What we've discovered, and we haven't had enough time to sort of look into why, we've discovered a couple things. One, sometimes when those agronomists are taking them, they'll take a 12-inch core, okay? They'll take a thing, stick it in and pull it out. It's maybe because they're doing mineral testing of the soil, right? Carbon or something. When we test microbes, right, they live around the rhizosphere. They don't live 12. I mean, there's some 12 inches down, but it'll be different. So they take these cores from different places. They throw it in a bucket, okay? And then they mix it up, sometimes with a shovel, sometimes with an auger, okay? We're testing living things, fungal content. You don't want to mush it up. And then who knows when they test it. If you're doing a chemical or mineral test, you can wait. But if you're testing living things, you want to treat them like living things, right? You don't catch a fish and throw it in the back of your truck for a week and then weigh it, right? And then certainly not eat it. So we say for our test, go get field moist fresh soil from the rhizosphere, test it right away. Don't let it dry out. Don't abuse it, right? Don't mix it up. We have anecdotally found that when composites are done, the results are very, very strange on the microbiometer. We'll find out a little bit more about it, but we don't quite know why that is.
And composite meaning from different areas.
Yeah, composite is taking it from different areas and sometimes different depths, mixing it all together, and then getting sort of an average, right?
So you want to do separate places. So Laura, what are some of the ways that people are using the microbiometer and having a lot of success with it? Why do you think?
Yeah, so a lot of people are using it, and the way we love to see people use it is to assess soil practices. So for example, I want to increase the mycorrhizal fungi in my strawberry population. So I'm going to put down an inoculant, right? Put down an inoculant, you can test. Some people want to test the input, but just because you have a jar full of microbes doesn't mean that in a month you're going to have those microbes in the ground. So what we tell people is test before, right? And then test depending on, when you think they're going to colonize, test in a week, 2 weeks, a month to see if those mycorrhizal fungi have colonized. Did your fungal to bacteria go up? Was it effective long term? Or was you put the mycorrhizal fungi down, but your soil was not primed to enable them to reproduce, right? So it's not necessarily that there was no mycorrhizal fungi spores. It's that you put them down and they died, okay? So, and if you have two fields, you can say, I'm going to inoculate one and not the other and see what that difference is too, right? Did I get a natural boost? Did I need the input? And also, how much did I need? These inputs can be very expensive. Is 1 cup per gallon enough, or is 5 cups per gallon necessary when I put it down? That could be a very big price difference. Same with cover crops. I want a cover crop that is increasing my microbes, right? I'm going to have 2 fields. I'm going to test one versus the other. So people use it really to assess what's going on with their soil and to compare their soil over time and place. People also try to use it to compare to other people, but that doesn't really work. We sell in 83 countries. The number you're going to get in Israel and Australia is not going to be the same as the rainforest in Washington State. It just doesn't, it can't get that. So you really want to have a competition with yourself to get the numbers up. And that's how people use it. And then, we also have a contingent of students who use it for science fair experiments, which is really cute. It's a totally safe kit. Kids can do it. And the app, they don't have to put in the information in. They can't join a chat group or anything. And that, we have a couple on our website, sample outlines, to test, if you have different pots with beans growing in it, can you see what soil grows them fastest, and the tea bag experiment as well.
Oh, I love that. I love how it's being used in schools. It brings soil to life, literally.
Yeah. And it's just a lot of people do it because they're curious and they want to know about it. And it helps them get more invested in understanding what they're doing, that there are real living things in the soil that they are stewarding through, right? That they're helping the microbes do better. The microbes are feeding us, like quite literally. I mean, the microbes in our gut, we would die without them.
Yeah.
And we're finding the same with the soil, right?
Right. Yeah, it really validates what we're practicing in our fields and on the land that we're stewarding. And then I just want to bring it back to the soil test with the kids, because it validates to them what they're seeing and witnessing, right, in a plant growth. And that's what I mean. It brings the soil to life for kids, and maybe even to farmers and gardeners too, which is so powerful because it's so invisible to the naked eye what's happening in the soil. But when you see, oh, that plant, only grew 3 inches and this plant grew like 6 inches, and what's the difference in the soil? And then you can actually see it and validate it through different visuals. It's just a different experience. So
Yeah, and it's very approachable, right? You don't need anything complicated to figure it out. I mean, unfortunately, we've lost a lot of our, a lot of us don't live on farms. We don't sort of live, I mean, I certainly don't know that much about growing, but there's a lot of information, and it's all different everywhere you are. Even microclimates have very different areas that things do well. So, I mean, it's such an exciting thing to be able to participate in, but it's not easy. It's not a one size fit all. And the recovery of soil will not be a one size fit all as well, right? There's not going to be, one device or input or practice that's going to fix it all. It's going to be lots and lots of little things that people do.
So Laura, your mom was really the scientist behind the microbiometer and soil testing, and you're actually an accountant, right? And you're continuing her legacy and doing incredible work. And I'm also curious what biodiversity means to you.
I mean, biodiversity is such a big word, right? I mean, it goes from what we think about all day, microbes, up to plants all over the world. It just means that there's room for all the things that nature wants to encourage to grow, right? That we are accepting both of different things in different places. Like, for example, weeds, right? I mean, a weed is just a plant that you for some reason don't want to grow in a particular place, right? So grass is a weed in a farm, but it's not a weed on your lawn, right? But a lot of the agriculture is now moving to more holistic, right? So there's more biodiversity, meaning you worry about the microbes, you worry about the plants, you worry about the animals on the plants, you worry about the birds in the sky, you worry about the animals eating the food, the other animals eating the animals, us eating the food, to sort of make room for all of those life forms, I guess. Sort of my attempted answer, even though I don't have a PhD.
Yeah, so true. Well, thank you for joining us today, Laura. Where can we find the microbiometer?
Oh, yeah. The microbiometer is on our website, microbiometer.com. I'll tell you that we are a US-based company located in Hudson Valley of New York State. We are currently an all-women company, which just happens to be an anomaly. But we do manufacture and ship everything ourselves direct to growers all over the world. So a starter kit's $135. It has 10 kits in it, 10 tests in it. You can order refills. And then our app is free to download. And that's how you read the results and store all your results.
And you're also on Facebook and Instagram at MicroBiometer.
Yes, yes. And I think that there's also a discount code for Heart and Soil
Right, Heart and Soil. Yeah. So it's Heart and Soil 15. You can get.
You can get a 15% discount on any order, and it's free shipping throughout the United States. So it should be pretty easy. Right. But thank you so much for having me. It was really fun to talk about soil testing, and I hope everyone listening got something out of it.
Yeah, thank you for joining us, and thanks for the work you do.
Okay, thank you.
You can learn more about the microbiometer at microbiometer.com or find them on Facebook or Instagram at microbiometer. And you can use the code HEARTSOIL15 to get 15% off of your very own microbiometer. Thank you for joining us. I'd love to know what soil tests you like to use and what questions you have about testing your soil and what your successes have been and what some of your challenges have been. Or maybe there's a soil test out there that you really want us to know about. Send us an email. Let us know at connect@heartandsoilmagazine.com. Make yourself an amazing day. Big love and bright blessings.